Right, so welcome to your very last chapter. And I wanted to note to you, if you look at the scheduling way down here at the end, I had mentioned that chapters 20 and 21 aren't actually part of the goals of the class. There it is right there. It says describe how genes are expressed to make proteins and how gene expression is regulated. That actually was chapter 18. So that formally was the end of the course there. But I mentioned I wanted to talk about these two chapters primarily because they're so important, particularly the biotechnology chapter. And I wanted to give you everything I thought was necessary to hopefully help you succeed in your future classes. So this is hopefully not just icing on the cake, but just that much more to help benefit you in the future. So there won't be a lot on the test that relates to chapter 21. So I'm going to kind of move through this pretty quickly, but I just wanted to give you an idea of what it was about. So you can see genomes and their evolution, particularly this evolution. You really are segueing now into Bio 2. Bio 2, again, starts with evolutionary theory, goes into the major organisms, so like the major phyla, and then finally into ecology at the very end. It's a change. Now, you use everything in Bio 1 and Bio 2. I regularly talk about Bio 1 in my Bio 2 class, but this really is that segue. And don't forget, evolution just means change over time. That's all that word means. So it's nothing nefarious or anything to be too concerned about. It's just have things change over time and how, and of course they have. So this is really kind of moving you that way. So we really looked at then in Bio 1, genetic theories, all that background stuff we did on cells and the chemistry, got you ready for genetic issues. And genetic DNA, genetic theory is really the underpinnings of biology along with evolutionary theory. So we really want to know then, what really is DNA? How does it work? Again, it's nothing magical. It's powerful, yes. It does two things, copy itself and makes the RNAs. But really, again, how does it vary from one species to another? You can see you and I and chimpanzee. And by the way, there's some talk about bringing the pan genus into the same grouping with a Homo genus, and both meaning human. I've seen some discussion with that in regards to anthropology, because really, what does it mean to be human? And again, phenotype comes up in like behavior. So really, what's the difference between these two? It depends on the research. It's anywhere from 1 to 2% on the DNA. I've even seen some DNA saying it's less than 1% between a human and a chimp. Escherichia coli, bacteria, obviously quite different than you and I or a chimpanzee, and brewer's yeast and corn and fruit fly. Really, again, the DNA of all of these guys. And that's really what this is, genomics. The entire DNA complement that makes up an organism. And so I've also said at the beginning of the semester, to be a good biologist, you really need to be good at two other things, chemistry and mathematics. You don't need to be an expert. You don't need to be a chemist. You don't need to be a mathematician, but you need to be comfortable with it, at least those levels that you have to do in your research. So bioinformatics really is the combination of those, the combination of your biological data, yeah, so your genomic information, your DNA, your genetics, along with computers and how you computationally understand the data. I had a friend in my master's program, brilliant guy. He was getting his master's in genetic biology, but at the same time, he went off on his own, learned like 3 computer programming languages. So he was the guy that not only understood the science, but also understood how to program the computers to analyze the DNA. It was absolutely well done. And he was like snatched up instantly into a PhD program, went and did his postdoc, and now he's a professor out in Alabama, I think, but he's doing quite well. So that's really what bioinformatics was, the combination, not just mathematics, but what computers can do in the study of genetics and therefore also genomics. So this is one of the more, in my opinion, for what it's worth, interesting projects. The Human Genome Project, notice this started in 1990 and took 13 years to get done. And so this was trying to figure out for a typical human, whatever typical means, what are the A's and T's and C's and G's that makes up a human? It's really what it was about. And then the second part of this project is now to figure out how it all works together. And that's the harder part. And the first part took 13 years, mind you. So this is challenging to say the least. And so obviously mapping everything out. So we'll look at this real quickly, linkage mapping, physical mapping, and of course, the A's and T's and C's and G's. So in regards to the linkage map, this is just looking at on a chromosome, where are these alleles and what do they actually code for? So like, oops, I have it out of order. Sorry about that. So here you go. You can see, maybe wings and the legs and the aristae. It's just kind of showing you where these traits are, these alleles are on the gene. And that's a little different than a physical map, showing you the distances between these markers. So again, you can look at that as well. So you go, here's a chromosome, and you can see the physical distances between these alleles, that kind of idea. So we're just mapping where these things are. And then looking to see the sequence and trying to figure out, again, the entire complement for an organism, which is billions of base pairs long, and sometimes even trillions of base pairs long in some regards. I've heard, what is it, 1,200 million base pairs in some plants, if I remember correctly. So it's really just quite amazing how much DNA some things can have. So this is one of the techniques that was first available, the shotgun approach. It was kind of like chopping up the DNA and then looking for overlaps, and with the overlaps kind of build up the entire sequence. Notice it used obviously computer programming in order to do that. So computers were obviously becoming a very important part of the science, the biology, and certainly our ability to probe into the world around us. Not surprisingly, people were skeptical that this was going to work, but it eventually gained acceptance, and they used it in the Human Genome Project along with other techniques to try to figure out, again, the typical A's and T's and C's and G's of a human, whatever that meant, including this one, metagenomics. Man, I wish I had this when I did my master's work. I had to go down and do seagrass and figure out like all the different times seagrass would change in a bed, where these guys were able to go down and take like a little sample of the sediment and figure out exactly what was in it. So that was amazing, where I had to like look under a microscope and for anything there, a worm, a seagrass blade, whatever it is, try to figure it down by the phenotype all the way down to the species level. It was incredibly taxing work, where they were using machinery to be able to figure out, at least in microbial communities, what was there. Microbial ecology is very important for understanding the environment. So it's really amazing how the sciences definitely changed. So databases were obviously set up so you can have like a repository of this genetic information and then go and access that and maybe do comparison. Like I have the sample, what does it compare towards? So that gets you into these banks and look at all these different, not just organizations, but even countries that have come together to create these banks. You should know the NIH, National Institute of Health. You might not have heard of NCBI, National Center for Biotechnology Information, but it pops up all the time in the sciences. Europe's obviously involved, Japan and China. It's just amazing, the collaboration in the sciences, including GeneBank, which I do have here. There it is. GeneBank, you can see here, it's a repository, genetic information. I actually remember the first time I came across a GeneBank. It's when I took genetics in 1993, and it was super exciting that you could submit your sequence of DNA, let the computers work on it, and then ping back saying, oh, I think it's for this, I think it's for that, I think it does this, and maybe protein, what protein might be doing, that kind of idea. So again, you can use that software to look for sequences, what the protein does, the amino acid stretches. It was really, I mean, this is 1993, mind you, really quite amazing. I can only imagine, including these 3D views. I mean, look at this. The data may not make much sense, but building up these 3D views, what you can do now in 2022. Remember, I'm a geobiologist. I'm not a geneticist, so I haven't kept up specifically in the field. I keep up in my field, but not supposed to this. So I can only imagine just how much is out there. What an exciting time it must be to be able to take genetics. Generally, after Bio 1, you go off to take Bio 2. After Bio 2, not uncommonly, you take genetics. So I can only imagine some of the most amazing things you're studying out there with this. I see it in the literature, but technology-wise, it's just a little bit outside my field. But still, it's really quite cool. So again, you're building up these repositories of genetic information and looking for things like, what does it code for? So that's what gene annotation is. DNA sequence is equal to this kind of coding. I think I have a quick little video to show you that. Let me change over my sound. Okie dokie. Hope you enjoy it.
In this video, I will be discussing the meaning how to find out why. Genome annotation is the process of identifying the location of genes and discovering their functions such as roles in the replication of DNA, as well as annotating other genetic features, for example, non-coding RNA from a raw sequence. This process can occur automatically or manually. This diagram I've created shows the process of genome annotation from a visual perspective. As you can see at the top, the genome sequence consists of a list of DNA bases, C, G, T, and A. Through the process of genome annotation. This DNA sequence is converted into the display seen below. From this, you can see much more about the position of key elements within the genome, such as the enhancer, mode site, the location of exon, and repeat region. This process is the only way to really understand the genome environment of an organism, because without it, you would not be able to identify any genetic elements from looking at its genome sequence alone. Now we have reviewed what genome annotation is. The next important step is to learn how the genome is annotated. Firstly, a genome of an organism is selected for investigation. This genome can either undergo automatic or manual annotation. Automatic annotation is performed through the MAKE pipeline, which requires no user input and is run through the computer alone, or the genome can be annotated through manual annotation. This is carried out by the scientists themselves through genomic programs such as the Apollo, Geogas, and Galaxies. This form of genome annotation is considered more reliable and accurate. Now we know all about genome annotation. Why is it effective? This diagram represents an old genome annotation compared to a newer, updated one. There are many situations such as these where the genome annotation is outdated and lacking recent information on the location of genetic elements. I think that problem with misannotation is seen in the diagram where an old annotation identifies one exon, when in the recent genome annotation there is found to be two separate exons. This can cause an accuracy of an input when there is thought to be one exon, and in reality there are two cousins. There are other types of misannotation in genome annotation. For example, large types of genes can be considered in the wrong place compared to where they should be, as seen in the figure presented. The study was carried out to identify the amount of misannotation which occurs within genomic databases. Normally, in the NR genomic database, approximately 40% of people with attributed were misannotated in 2005, and as this is increasing with time, it is considered to be a much higher number in 2020. The number of misannotated genomes that are being published to genomic databases are increasing rapidly. This is causing a growing issue that can affect numerous genomic experimental research. Therefore, recent genome annotation of organisms is essential, and we must be doing everything we can to ensure this problem does not continue to grow. Thank you for listening to my video about genome annotation. I hope you
There you go. Gives you an idea then of how the process works, some caveats to it. We still have more to work on, of course. So a lot of science left to learn. So yeah, it's a lot of computers building up this kind of information and letting us compare genes we know, our genes to other species. And of course, we do that all the time in regards to comparison of, what makes a human versus a chimp versus another organism. And I say this a lot in my bio 2 class, but a human is a human is a human. Bear in mind in the sciences, we don't recognize races. That doesn't exist in the sciences and biology. A human is a human. So we compare humans to pan troglodytes, a common chimp, to maybe a silverback gorilla, then outside the primate lineage, maybe to a cat, like my little kitten that's sitting right here, that kind of idea. Proteomics is a similar idea, but this is just a study of proteins. Remember, the output of DNA is the RNA. By making the RNAs, you make a polypeptide, and your polypeptide might be a protein. So again, it gives an idea, kind of working backwards to what the genes do. Another one, of course, is for illness. It's kind of intriguing to see when this was made. The Cancer Genome Project, they're noting, is comparing gene sequences, looking for, what typifies some of these cancers, maybe being preventative, maybe treatments. They're noting that it's been working, so they want to apply it to more than, what did they say, 3 cancers here. So when you look at it, oh my goodness, has it been productive? This was a Human Genome Project. Oh, there it is. This is the Cancer Project. I didn't show you this one, but the Human Genome Project is out there. You can go and study it if you want to. But this one is the Cancer Project, the Cancer Atlas Project. And look at all the cancer types they have now. So it's really branched out, looking at these cancers and trying to figure out, why it happens and how to predict it and how to work with it. As I said, I think last chapter, we're getting better with this and finally getting to the point where we're talking about cures as opposed to just, the 12,000 known human illnesses. So, 12 years ago, almost 13 years ago now, we've already had a lot of genomes that were sequenced, a lot of bacteria. This is the everyday bacteria. This is the extremophilic bacteria. This is the plantae, fungi, animalia, and the protistans. And so we're still working on it and still working where we go. Now, these is just a general idea of how big some of these sequences could be. Notice this millions of base pairs. So especially coli, 4.6 million base pairs. And you can go down the list. Here's Drosophila melanogaster, that's a fruit fly, 165 million base pairs. Corn, 2.3 billion base pairs. A mouse, 2.6 billion base pairs. Homo sapien, 3 billion base pairs. But look at this one, the lily plant, 124,000 million base pairs. My goodness gracious, that's what I was talking about, like getting towards the trillions. It's just incredible. Why in the world a plant needs that much DNA? Who knows? But it was what works for them. Now, I was noting to you, I think last chapter as well, you can see Homo sapiens, we have roughly 21,000 genes, roughly the same as a mouse. Again, that's neither good nor bad. It's not a plus or minus to either species. It's just that idea, again, you could do a lot with a little, as we were noting with the alternative RNA splicing. So pretty cool. So there you go again. Again, it's variable, you know, how much is out there. You can't really correlate the number and size of a genome with complexity. It just doesn't really work that yet easily. So there you go. Number of genes is not correlated with genome size. It's just not quite that simple. We're still trying to work out how many humans have. And then again, of course, there's your alternative RNA slicing I just mentioned, allowing you to do a lot with a little. So as I mentioned to you, most of what DNA is non-coding DNA. It used to be called this junk DNA. Never liked that phrase because it kind of, in essence, says you throw it away. No, now we know most of it is non-coding, and very little amount of DNA is actually coding DNA. So again, you can see here, 98.5% does not code for proteins, RNAs, or tRNAs, or RNAs, mind you, or tRNAs. It just simply doesn't. So 25% then codes for introns and gene-regulated sequences. So 25% codes for introns and gene sequences, regular sequences, only 5%. So again, it's not a lot that codes. It's just kind of intriguing. There is both pseudogenes, which have mutational issues, and because of that, they don't function anymore. Repetitive sequences could be good for complexity if they're transposons and they jump around. That could be deleterious. So again, there's a lot of complexity here. There you go. Only about 1.5% of coding DNA is exons. Only about 5% are introns. Again, that's what I was trying to say here. Again, most of this is non-coding DNA. Again, it just kind of is what it is. It's not junk, though. It's very rare that nature keeps things that it doesn't need. So a lot of it could be regulatory. Some of it we're still trying to figure out, like in here, what does it actually do? And that's okay. So Barbara McClintock, who, if I recall correctly, and I could be wrong, but if I recall correctly, won a Nobel for this. She was working on Indian corn and showing these transposable elements. These are like jumping genes, and that they could be parasitic genes. So when they move, they can cause deleterious issues to crop up, genetic diseases to crop up. So there's two ways they could do it. Transposons, notice they're moving by DNA, or retrotransposons, which move by RNA. But either way, they're moving, and they might cause problems. So there are two I just noted back here, the L1 and the ALU. We're still trying to figure out what they do. So you can note that here, primates ALU, and you can see that they're transcribed in RNA molecules. But like I just said, we're trying to figure out what they do. And humans use this one, the L1, the line one. Notice not much transposition, so may not hurt so much. It actually might help with gene regulation. So intriguing, especially when you're differentiating like chimpanzees to humans, intriguing some of the differences. Multi-gene families are groups of genes that code for something similar. Here they're noting alpha and beta globulins. Again, they're just expressed at different times in like your developmental stages. So it's just a group of genes that code, you know, a little bit about something like similar kind of product. So duplication, rearrangement, mutations, they all kind of go together. Mutations, again, be careful with that. Mutations are just change of the DNA, like we said many times. It could be good, it could be bad. A lot of mutations are silent mutations, but I've read the vast majority of them are silent mutations. So we just need to be a little bit careful here. Duplication can lead to complexity. Complexity can lead to good things, but again, rearrangements and duplications can also lead to bad things. So this is why we have a note of caution with mutation. While it is one of the driving forces of evolution, just let nature take its course. Don't go out and try to get a mutation or something like that. Again, even Charles Darwin noted that evolution happens on the populational level, not the individual level. Yes, Dawkins questioned that in the latter 20th century, but it's complex, and therefore what helps, what hurts, it's really hard to know. But all these together do allow for evolution to take place. And that's kind of where this chapter is going to end up. So not surprising then, the very earliest life didn't have much going on. And then over time, genome size increased. I mean, look at the lilies again, 1,200 million base pairs. Oh my goodness, who would have thought? At least I think that was a lily, right? Who would have thought that a plant, yeah, the lily family, would have so many genes, but they do. So again, it just got more and more complex as we went. Natural selection obviously will work on that. Remember, natural selection works on things that's already there. Natural selection, therefore evolution is like editing. It doesn't create, it edits. You already have it in the gene pool to see how it functions. Does it confer an advantage or not? And to be biologically successful means to pass your genes on. So is it helping the population or not? So again, we got to kind of see what it does in regards to the evolutionary issue. So duplication issues right here, accident in meiosis, non-disjunction, for example, where you get the N+1, N-1 condition, a little bit too much DNA, a little too light DNA, that might lead to genetic problems, trisomy 21, Klinefelters, whatever it is for humans, polyploidy conditions. I see this being a little bit more malleable in plants and maybe the animalia, but it can lead to certain problems with the animalia. So these extra set of chromosomes, I remember the plants, man, they could be 3N, 4N, 5N, 6N, depending on the plant, or animals, particularly mammals, really can't do that. But these are accumulation of mutations. And again, they might help the organism, but they may not help the organism. So if they persist, maybe it's conferring advantage. It might help them survive, might help them reproduce. If it doesn't persist, then probably gets dropped out of the population. So even with humans, we could talk about this. There's a lot of words, sorry about that. But even with humans, we could talk about this. Humans and chimps had a common ancestor somewhere about 5 to 7 million years ago, somewhere in there. And then chimps went their way, the pan line, and Homo eventually went its way. But we all had these 24 pairs, we think. And then somewhere along the way, humans had a merging of chromosomes. And we went from having 24 pairs to 23 pairs. So again, this like merging of chromosomes took place. And so we're looking for that, why did it happen? Why was there this divergence between humans and chimps? And then again, this fusion that happened with Homo sapiens, it didn't happen in the pan line. Of course, we're looking into that. We can correlate that with evolutionary history, like right here, they're noting again that with mammals were kind of like these smart, almost like, I don't know, like iguana kind of organisms running around rocks, the amnios, mammals and what are called reptiles, although taxonomically that word isn't really valid anymore, but we were together in the amnios. We had a common ancestor, and then if I can still use a word for today, reptiles went their way, the mammals went their way. They're actually diapsids, better word for them for the reptiles, and synapsids for the mammals went their way. But again, mammals really didn't get their chance until the major dinosauria died out. Of course, the birds are still there, and that opened up like niches. When niches opened up, mammals, flourished, and here we are, 65.5 million years later, having this wonderful little discussion. So we talk about that, these changing in the environments, allowing for diversification, these niches. Niches, your fundamental role is what you do in the environment. No two species can occupy the same niche at the same time. So we talk about that. Those niches opened up and we flourished, and maybe how that affected our DNA and allowed for this recombination events to occur. Therefore, to some lead to advantage, to some lead to disease. And who knows what diseases? There could have been diseases that have dropped out. We don't talk about them anymore. Maybe never even knew they existed. That kind of idea. So still a lot of research to be done on that. So prophase 1, again, I really harped on that when we talked about it in meiosis. That was like that really one unique thing in the meiotic process that you don't see in meiosis 2, and you certainly don't see in mitosis. So maybe something happens there that allows for that N+1, N-1 condition, that non-disjunction, and your chromosomes aren't separating correctly. Maybe genes are jumping, and you get crossing over between these non-sister chromatids. I made light of that back in meiosis, saying that's not usually a good thing when non-sister chromatids swap genes. So again, something might be going on. And then, of course, as you have these changes, genes can end up with very different functions. Here they're noting again that they're encoding for two very different things, lysosomes versus lactalbumin in mammals. And so this one, where it might have come out of the same thing originally, perhaps, one is now being used to protect you from bacterial infections. The other one is dealing with milk production. By the way, as a side note, because again, I don't know who you're going to have for your bio2 teacher, bear in mind, production of milk is not a specific trait of mammals. There's two things that make you a mammal: mammary tissue and hair. Not specifically making milk. There are other organisms that make milk-like substance for their young that are not mammals. So be careful there. But here again is one where you get a differentiation that happens from a gene duplication. So again, we got to track this and find out what was it originally, what did it become, is it helpful, is it not helpful, that kind of idea. So you can get duplication issues, you can get shuffling issues, duplications could lead to complexity. Shuffling might lead to new and novel functionality, but then again, if they're jumping genes, it might be parasitic genes. So there's a lot of issues that might crop up here. And with exon shuffling, notice these errors, matching of exons, again, it could be non-allelic genes, maybe problems arise, this can be quite problematic. So again, kind of be careful there. And these transposable elements, again, are getting back towards like those jumping genes again. So take the slide and connect it way back here. Just so I can make it, there we are. So the transposons are jumping by DNA, where the retrotransposons by RNA. Back to where we were. So again, how are things moving around? You get these copies, they get inserted. Once they're inserted, are they helping? Here they're blocking. So again, what are they doing? Do they cause a new feature to arise? Do they cause a problem to arise? So again, you have to see, but notice more times than not, it can lead to something pretty bad happening. So usually detrimental, but might prove to be helpful. You have to see if it's detrimental. The individual that just happened to have it probably doesn't, who knows, maybe they don't even make it to gestation or incubation. Maybe they're born, they can't reproduce. Maybe they just can't pass their genes on. Maybe they don't compete well, so they can't get resource. And in that case, it's not advantageous. If it is, it would be detrimental in that case. If it is advantageous, then maybe it helps them to get resource better, maybe attract a mate, pass their genes on, that kind of idea. You just kind of see. And therefore, does it accumulate in the population or does it fall away? So we've been doing this for a while now, 25 years. That's a nice chunk of time to be kind of coalescing all this data. We're beginning to get an understanding of the history of life on Earth. I think in this textbook, in chapter 25, you actually have to learn the geological calendar. It's very important to know, the chronology of this planet, what we think has happened throughout Earth's history, both in regard to the Earth itself, but also life on this planet, and therefore, rates of evolution, how often things change, why they change, and therefore getting this kind of morphological diversity. Be careful morphology. Morphology, like even a body plan, can really fool you. There's kind of three ways to look at evolution: morphologically, biochemically, and DNA. Now, biochemically, that might seem like, wait a minute, isn't DNA biochemical? This biochemical might mean like protein analysis, and then of course DNA. All three are important, but man, morphology can really fool you, where you think two things are really similar based on morphology, but they're actually not. So you got to be careful, and you'll see lots of examples of that in bio too. If I had to pick one of the three, just like the one to maybe default to, I'd probably pick not so much morphology, although I'm not discounting it. I'd probably pick a genetic comparison, DNA comparison among species. And that's what this one is, genome comparison of closely related species. So like human versus chimp, that kind of idea. I think this author notes 1 to 2%. Whereas some authors even note less than 1% difference in the DNA between a human and a chimp. Although morphologically, I don't think you're going to confuse a human with a chimp, that sort of idea. So a closely related species, obviously, that's recent events. 5 to 7 million years ago, that's not really that long ago, especially when life on this planet's been here for 3.5 billion years. But maybe then you maybe want to do, say, Homo sapien versus Homo neanderthalensis. So what was a Neanderthal? Was a Neanderthal Homo neanderthalensis? Or were they Homo sapien neanderthalensis? Well, Neanderthals died out, depending on the research you read, about 30,000 years ago. That's definitely a recent evolutionary event. Or we could talk about very distant events. Maybe the separation point between, say, reptiles and mammals, that kind of idea. Or even within the primates, monkeys, and we call them colloquially great apes, although that's not taxonomic. But that was, what, 25 million years ago. So that's much more distant separation. To do that, we use these trees. These are called phylogenetic trees. This is the ancestor, and that's like a speciation event. It's called a node. There's a node. If it goes all the way to 0, these would be extant species, meaning they're still alive. If something doesn't make it to 0, that means it's extinct. Well, archaea is still here, the extremophilic bacteria. Eukarya, in regards to the protists, the plantae, the fungi, the animalia, are still here, and of course, everyday bacteria is still here. But they're noting way back here that everybody had a common ancestor, and then there's a speciation event. And then bacteria went on their way, and I even made reference to this in a previous lecture, that the archaea have some cases more similarity to the eukarya than they do to the bacteria. But that's still 3 billion years ago, we had a common ancestor. And then here, focusing on the eukarya, obviously much more recent, you see the common ancestor, then a split point here, a node, and humans and chimps went their way, what eventually became a mouse went its way, and then even here, the common ancestor to the human and the chimp, again, 5 to 7 million years ago. and you can talk about that node, and these might be like sister taxa, that kind of idea. So you'll talk about this a lot more in Bio 2. These phylogenetic trees come up all the time in Bio 2 in relationships. So you can compare both species that diverged recently as well as a long time ago. And so, especially a long time ago, you want to look at genes that just don't change very quickly, especially when you're doing this. I mean, you're going back billions of years. You want to look at something that's hung around a long time. So we look for those genes. We look for not just those genes, but an organism to model and see what can maybe be used. So here's that data I was talking about with chimps. I have to admit, I was a little surprised with this author taking the stance that humans and chimps differ by 1.2% at a single base pairs and 2.7% on insertions and deletions. Okay, it seems a little high to me. Again, I've seen some authors generally note about 1% difference between humans and chimps, and I've even found a couple of papers now where it's less than 1% difference in the DNA between humans and chimps. Either way around it, I mean, it's not much difference. Statistically, we're pretty much the same thing, but again, morphologically, you're not going to differentiate. You're not going to have difficulty differentiating Homo sapien from a pan-troglodytes, a modern human from a chimpanzee, a common chimp. So it's just amazing the power of DNA. Again, it's not magical, it's just powerful. That's all. So in that case, then we look at some of the differences, and we went our way, chimps went their way, and you can see some of the differences. How we deal with diseases. Maybe we came across things that they didn't. For example, HIV is still tough for us to deal with, or SIV has been around a long time in the chimp population, and I've read it's nothing more than like a common cold for them now. Or regulation of brain size, but be careful, big brain doesn't mean smart. It's the neocortex and paleo cortex we look at now. Again, those are transcription factors we talked about before, the homeotic genes, like the Hox genes, that kind of idea. This is kind of a neat one, the FOX, P2 gene for vocalization, where we obviously have very complex speech. Chimps, not as much, although they can learn things. How they learn might be a little bit different than you and I. So there's some neat research going on there. And pretty much last but not least, one last time, there's the homeotic genes, the homeo box. And with these are genes that control transcriptional factors. So how many things you're going to have and where you're going to stick them. These are transcriptional factors that deal with that. There you go, transcriptional regulation. I've talked about the Hox gene a couple of times. In the arthropods and the chordates, Hox genes come up a lot. Chordates would include things like you and I, arthropods, insects and chelicerates and crustaceans and those kind of guys. But again, how many arms and legs you're going to have and where you're going to stick them, that kind of issue comes up. So, okay, why not? So again, in the arthropods versus chordates, mammals are a kind of chordate. So again, complexities popping up, how many things you're going to have and again, where you're going to put them. And I'm not trying to say one is more complex than the others. Insects, by the way, are pretty darn complex on a lot of things they do, including their behavior, but it's kind of a neat comparison. And then, of course, you can talk about what's conserved, what's not conserved. Again, it's pretty much the same thing in regards to the Hox genes and expressional controls. Here to talk about legs, or how many legs you're going to have. Maybe you differentiate a fruit fly from Artemia salinus, which is brine shrimp. So again, fine, this is brine shrimp actually here. What do you know? So how many legs you got and where you're going to stick them versus say the grasshopper, how many legs you're going to get and where you're going to stick them. So it's just intriguing to see how these homeotic genes work. And it's not just in animals. Plants are very complex, and plants never get their due, which is unfortunate. They're obviously quite important to life on this planet, quite important to you and I, food and medicine and textiles and clothing, you name it. Plants are incredible. And they have their own version of these Hox kind of genes, the MADS box that they work with. So don't discount your plants. I know I've spoken about it a lot this semester. Hopefully your Bio 2 teacher will talk about them a lot as well. And with that, you've made it to the very end. Congratulations, folks. I hope you've enjoyed Bio 1. Again, any questions you might have, feel free to let me know.